Hi there, this is Mr. Cho, and today I'm going to be talking to you about fats and proteins. Let's look at this picture. So you can see some salmon, some fish oil, almond, nuts, avocado, oil. Now these are all fats, right? Fatty foods. And fats are also known as lipids. So the chemical composition of fats is carbon, hydrogen, and oxygen. This is similar to carbohydrates, but in a different ratio. The sources of fat include butter, cheese, nuts, and oils, and they can come from animals or plants. The proportion of the carbon, hydrogen, and oxygen are not fixed. They can vary from fat to fat. Now, what is a fat molecule made up of? Okay, basically it's made up of four things, but two things: glycerol and three fatty acid chains. So glycerol is this. You can say that it's the backbone of the fat molecule, and there are three fatty acid chains over here. Okay, so different fats can vary. The length of the fatty acid chain can vary. The shape of the fatty acid chain can vary also. But all you remember is that a fat molecule consists of one glycerol and three fatty acids. Now, if you've ever touched, some oil or fat before, you know how it feels like. It feels kind of smooth, right? Now, where does that smoothness come from? It actually comes from the glycerol. So glycerol feels smooth to the touch. And you have seen fat before, you've seen oil before. You know how it looks like. It looks a bit yellowish. The yellow color actually comes from these fatty acid chains. Now, this is some FYI information. This will not come on any test. Basically, what are saturated fats, unsaturated fats, and trans fats? Now, I don't want to go into too much detail, so all I'm going to say is that saturated fats are bad, unsaturated fats are good. Okay, but unsaturated fats sometimes can be converted into trans fat by overcooking or extremely high temperature cooking, so it can become bad again. So saturated fat bad, unsaturated fat good, trans fat bad. Okay, so if you see this kind of thing in the food, don't eat. Okay, where to find unsaturated fat is from olive oil or fatty fish such as tuna, salmon. Alright, what are the functions of fat? There are four main functions. The first is that it's a source and store of energy. Now, if you recall about the functions of carbohydrates, you know that the function of carbohydrate is primarily it's an immediate source of energy. Okay, so that means our body will use carbohydrates first. If, for example, or somehow your body runs out of carbohydrate, then it will eat into the store of energies, which is your fat. Now, fat actually stores 2 times more energy per gram than carbohydrates. That means that fats are there's more energy within a unit of fat compared to carbohydrates. But we can only use this after our body has finished using the carbohydrates. So when people want to lose weight, right, they want to lose their fats, they will go for very intense workout or very long workouts, running for many kilometers. What are they doing? They want to use up all their carbohydrates in the body. Then they want only after the carbohydrate is used up, then the fats will start to be used. Okay, so the second function of fats is that it's the main component of cell membranes. Now, if you look over here, now this is a picture of the cell membrane. As you can see, it is a layer of fat, and this is one of the reasons why it is a partially permeable membrane. Okay, third function, insulate the body from heat loss. Let's look at this cutie pie over here. It's a seal. And why does this seal have such a thick layer of fat? It's called blubber. Take 10 seconds to think about it. The answer is because fat is an insulating material and it helps to reduce the loss of body heat as an adaptation to the cold environment. So this seal is adapted to its environment. It has lived there for many years, many generations. So it has its body has evolved to have a lot of fat to protect it from the cold. Fourth, fats protect our organs from damage. Okay, this means that, for example, our heart has a layer of fat around it, and as well as the other intestines, our small intestine, our stomach, there's a layer of fat around it. Why do we need it? Okay, it actually protects our organs from damage. Now if you think about two boxers, punching each other, why is it that they are not severely damaged? Okay, the reason is two reasons. First reason is they have muscles. Of course, they build up their muscles. And second reason is because they have fats to protect their organs from damage. Fat is a good shock absorber. Okay, how do we identify fats? Okay, we will use this thing called ethanol emulsion test. So, a cloudy white emulsion will be formed when ethanol and water are added to fats. An emulsion is a suspension of small drops of a liquid in another liquid. Okay, so a suspension means that it's not being dissolved. So actually what you see here is emulsion. You will see small little drops of liquid in another liquid. How do we do this test? Okay, for liquid foods, we will add 2 cm³ of ethanol to a drop of coconut oil in a test tube and shake the mixture thoroughly. Then we will observe for a while. After that, we will add another 2 cm³ of water to the mixture and shake the mixture. Then we should see the cloudy white emulsion. Okay, so we'll do 2 cm³ of ethanol, then 2 cm³ of water, then we should see the cloudy white emulsion. Okay, for solid foods, we'll crush the peanut into small pieces and place the pieces in a test tube. We will then add 2 cm³ of ethanol and we will shake it thoroughly. After that, we will allow the solid particles to settle down, and then we will decant the ethanol, which means we will pour off the top layer of ethanol into another test tube containing 2 cm³ of water. Okay, we don't want to get the peanut particles into the second test tube of water. Okay, we should also observe a cloudy white emulsion. Let us watch the video of ethanol emulsion test.
The ethanol emulsion test is used to test for the presence of fat. First, add a few drops of coconut oil into a test tube. To the drops of coconut oil, add 2 cm³ of ethanol. Shake the mixture thoroughly. Next, add 2 cm³ of water to the mixture and shake the mixture. What do you observe? If fat is present, a white emulsion develops. If fat is absent, the solution remains clear.
Okay, hope you enjoyed that video. Now let's move on to proteins. Looks good, right? All this food. Okay, now there's some fishes, chicken, eggs, but do you also know that there are a lot of vegetables that are actually high in proteins, such as broccoli? The chemical composition of protein is carbon, hydrogen, oxygen, and nitrogen. So again, similar to carbohydrates and fats, but with the addition of one more thing, nitrogen. And this is a very important part of proteins, nitrogen. So where do we get proteins from? We get it from nuts, milk, eggs, and meat mainly. Okay, it's composed of the elements carbon, hydrogen, oxygen, nitrogen. Alright, what is the structure of proteins? The basic building block of proteins are amino acids. So amino acids form the building block of proteins, and there are actually 20 different types of amino acids that occur in nature. Okay, so this is just a diagram of amino acids. You do not need to know this, but what I want to point out is that there's actually a little acidic group over here, and there's a different group here called R group, and there are different combinations that allow you to create different amino acids. Okay, and then when you add all these amino acids together, you form a polypeptide and then a protein. Okay. Excuse me. Amino acids are linked up by a peptide bond, okay, to form polypeptides. And these peptide bonds can also be broken by enzymes. Okay, so I've been telling you about enzymes. They break things down. So the enzymes actually break the peptide bonds. Now, when two or more polypeptides combine together, they form a 3D molecule called a protein. Now, why is it important that a protein has a 3D shape? Okay, if you think about enzymes require a specific 3D shape, right? And they have an active site which is complementary to the substrate. The substrate is going to also have a shape which is able to fit into the enzyme, and that is called the lock and key hypothesis. Okay, so all these drawings, these are all just 2D drawings, but the real enzyme will sort of look like this as a 3D representation. How do we test for proteins? We will do the burette test. The burette solution is a blue solution that is made up of sodium hydroxide and copper 2 sulfate, and it will turn violet or deep purple when proteins are present. Okay, how do we do the burette test? We will add 2 cm³ of sodium hydroxide solution to 2 cm³ of egg white solution in a test tube and shake it thoroughly. The name of egg white solution is sometimes called as albumin. Second step, we will add 1% copper 2 sulfate solution drop by drop, shaking after each drop. Okay, so let's just watch the video to understand how this test works.
Proteins can be detected by the burette test. To 2 cm³ of egg white solution, add burette solution drop wise. Shake after every drop is added. What color changes do you observe? If proteins are present, a violet color develops. If proteins are absent, the solution remains blue.
Okay, now in that video, they already made a pre-made burette solution. So that means they already had sodium hydroxide with copper 2 sulfate. But for our school, we would actually separate it out. So we have sodium hydroxide separately. Then we will add 1% copper 2 sulfate solution drop by drop. Okay, what are the functions of proteins? There are 3 functions. It's firstly, synthesis of new protoplasm for the growth and repair of worn out cells. Second, synthesis of antibodies. What are antibodies? Now, our body has white blood cells, and white blood cells help to defend our body from bacteria, viruses, and other pathogens, bad things. So there are a few specific white blood cells that can produce antibodies. Antibodies are proteins that are actually kind of, in a sense, like, let's say, homing missiles. Okay, they will home in on the target and they will destroy the target. The target being the viruses, bacteria. Third function of protein, synthesis of enzymes and some hormones. Okay, so we have talked about enzymes. We know that enzymes are made up of proteins, and that is why when you heat up the enzyme too much, or when the temperature is too high, the enzyme will become denatured. This is another 3D representation of an enzyme. It's a 2D representation of the certain hormone called human insulin, which is important for controlling our blood sugar level. Now, question: why do bodybuilders often go for a high protein diet? Okay, so fun fact, when you go to the gym, right, you do a lot of push-ups, do a lot of weights, you are actually tearing your muscle cells. You are tearing your muscle cells, you are introducing small little breaks in the muscle cell. And your body will then synthesize new protoplasm for the growth and repair of worn out cells. So your body will produce more muscle cells so that you will firstly fix the muscle, repair the muscle cells, and you will grow even more muscle cells. And that is why bodybuilders need a lot of protein so that they can build more muscle cells and repair their worn out muscle cells. Okay, this is it for the theory of this chapter. Hope to see you soon. Remember to press like and subscribe. Bye guys.